Good morning, students. Today class we are talking about bacterial vaccine. Okay, so during the session, in every pathogen, majority of the bacterial pathogen, you must have covered a part of vaccine or the prevention part. So this is a in total this is a combined class of the all the till now all the important pathogen what we have covered during this semester. So the learning objective of this lecture is important bacterial vaccine, what are there, what are the routes, and what are the doses of inoculation, and what are the indication. You know that there is a good list of bacterial vaccine, among what the important which are in red are already in the IAP program: the Mycobacterium tuberculosis, Corynebacter diphtheria, tetany, Haemophilus influenzae, strep pneumoniae, Neisseria meningitis. Even bordetella pertussis we have a component in one vaccine which I will cover. The other vaccines which against the Bacillus anthracis, Staphylococcus aureus or Yersinia pestis, they are still under development. The Salmonella typhi, Vibrio, Cholerae and Shigella, these vaccines against these pathogens are available and they are specifically used when it is indicated, or if the country is endemic, or if there is some outbreak, or you are traveling some area which is endemic, then these vaccines are indicated. So how these vaccines are usually prepared, or what are the targets for this vaccine, how it has been chosen? If we broadly, if we will see the bacterial morphology, then we know that the bacteria, majority of them, they have a capsule, they have a cell wall, majority of them they are covered by polysaccharide, they are long flagella, pili, and there are some outer membrane proteins or the adhesive molecules, there are DNA, and they release some toxins. Okay, this is overall if someone asks you what are the components of a bacteria, so these are overall the components of bacteria from which various targets are decided which can be utilized for vaccine for that particular organism. Okay, so when we compare the whole organism, sometimes the entire organism is taken to prepare the vaccine. When this kind of scenario happens, we usually attenuate the viral part or the bacterial part so that it decreases the virulence, but the antigenicity there it stays, or it can be inactivated. Okay, there may be attenuation or inactivation of the different components of the bacterial or viral part, and thereby it leads to an inactivated bacteria which lost its pathogenicity and retains its antigenicity. Sometimes they use the purified macromolecules such as the polysaccharide, the toxoid, some antigens which can be recombinant, and the vectors. Vectors can also be modified. It is known as recombinant vectors. We may target the DNA also. We may target certain protein, okay, synthetic peptide, and there may be multivalent subunits taking different units of the bacteria. Suppose we are taking the whole organism for vaccine. So as I told, they are either inactivated or attenuated, and for example, if we will consider the bacterial cells, anthrax, cholera, pertussis, plague, Yersinia, these are usually inactivated and the entire organism is taken for the production of vaccine. Tuberculosis, it is a live attenuated BCG. I will come to it Then typhoid, it is also a live attenuated bacilli. Viral particles, if we will take hepatitis A, influenza, which are inactivated. Measles, mumps, these are live attenuated. Polio, Sabin and Salk vaccines, these are also inactivated. Similarly, all other. We may take for examples of purified macromolecules. Suppose the toxoid. What is toxoid? When the toxin which is released by the bacteria, that is inactivated, it is known as toxoid. So diphtheria produces toxin and the toxin subsequently inactivated to toxoid. So these are inactivated exotoxins. Similarly, Clostridium tetani, or in the disease tetanus, they produce toxins and this subsequently inactivated. Similarly, we can take the part or the subunit of bacteria, for example, Haemophilus. Okay, Haemophilus influenzae, we take the polysaccharide and we add a protein carrier to it so that it can be easily transmitted, as well as it can mount the antigen, antibody or the immune response properly. Similarly, Neisseria meningitis, we take the polysaccharide. In strep pneumoniae, we take the polysaccharide, and in hepatitis B antigen, we take the surface antigen, and it is again modified as a recombinant surface antigen. So this classification or these examples are very important. You need to remember this so that you can able to answer the questions. Coming to the BCG vaccine, BCG stands for Bacilli calmette including. It is specifically administered intradermal injection. It is a live attenuated vaccine. As I told, it is live attenuated BCG. So it is developed by Calmette and Guerin in 1921, and the strain of Mycobacterium bovis, it is attenuated 239 serial subculture. So this strain, how it attains its attenuation by 239 times serial subculture in a glycerin potato medium, glycerin bile potato medium over 13 years. Then the strain which we landed, that is the strain ideal for the vaccination. Following vaccination, tuberculin negative recipient converted into positive reactor. The immunity for this vaccine lasts for 10 to 15 years. It is similar to natural infection except that it doesn't carry any risk of disease due to reactivation. That's why it is preferred as the vaccine for or against tuberculosis. The RD1 region of Mycobacterium tuberculosis genome is missing in the BCG. That's why it's lost its pathogenicity. The original strain was formed. This is the vaccine vial which is reconstituted with distilled water just before the inoculation. So it is originally made in the Pasteur Institute and gradually distributed in many countries. But when it was distributed, it was found that the daughter BCG strains which are attenuated from or which were subculture from the main the original BCG strain were differed widely. How did they differ? And so when it was analyzed, they fall into two groups, group 1 and group 2. Group 1, which is a Brazilian strain, and group 2, it is known as the Pasteur strain. It is mainly group 1 is a weak strain and group 2 is a stronger strain. Okay, so these are the difference. It is not needed to remember these differences. However, you must know that from the original strain, when BCG and daughter strain was distributed. Globally, there were different strains spread wide. Okay. So as I told, the protection rate, it varies from 0 to 80%. A major trial was done at Chengalpattu, Tamil Nadu in 1968 on this vaccine. It is the largest BCG trial in the world. So it involved around 2,60,000 participants. Children less than 1 month were excluded from this study to compare the posture or the Danish strain or the two different strain. It was conducted in an area with high prevalence of environmental mycobacter. Companion trial in North India not initiated due to political unrest, and in 1979 discouraging results, neither the vaccine imparted any protection against pulmonary TB was concluded. In Chengalpattu trial, the conclusion, these are the conclusions which were gained. It showed around 60% efficacy. It didn't reveal any protection in adults. 15-year follow-up from some protection in young person, especially against meningitis and miliary tuberculosis. This is a very big point what they got or they ended in from this big trial. Variation due to difference in the prevalence and virulence of the tubercle bacilli, the atypical mycobacteria and potency of the vaccine. There are variation in results. What are the complications of this BCG vaccine? This is one of the most important slide, and when you go to periphery to do some government job, you find that this is the most common thing after post vaccination one person can complain, and these are the complications which happens due to improper immune response or improper inoculation or something. So local abscess, this is the most important thing what happens after BCG inoculation or the BCG injection if you have not administered properly. So BCG abscess, indolent ulcer, keloid, tuberculosis, confluent lesions, leukoid lesions, lupus vulgaris, and there may be regional enlargement and suppression of draining lymph node, general fever, mediastinal adenitis, erythema nodosum, keloid formation, etc. Efficacy, it may not be protective against TB but protective for infants and young children against meningitis and disseminated disease. This is a, as I am repeatedly telling, this is a very important point, that's why it is given at the birth, that it prevents meningitis as well as disseminated disease, but it also provides protection against leprosy and leukemia. The recommendation in India, it just after birth, intradermal just before 12 months if missed the first dose, and should not be given after 2 years, also in children with active HIV. Babies from AB sputum positive mothers should not receive BCG vaccination just after birth, but after a course of preventive therapy. Coming to the strep pneumoniae. Strep pneumoniae, you know, it is a capsulated gram-positive diplococci, and the types, serotypes are distributed from 0 to 90, and this immunity against this streptococcal pneumonia is type specific, that is every capsular type has different immunity. So antibodies against this capsular polysaccharide is actually raised when there is infection. That's why they targeted this capsular polysaccharide as a part of vaccine. So out of these 90 serotypes, they found that around 23 serotypes which are prevalent worldwide and which are targeted for the pneumococcal vaccine. So this 23 capsular polysaccharide vaccine, it provides around 80 to 90% of the protection and the person with enhanced risk, who are the high risk person, the person who was having absent or splenic dysfunction, celiac disease, chronic lung disease, chronic heart disease, diabetes mellitus or HIV. These high risk person should be given this vaccine definitely. It is not recommended below 2 years. Now why it is not recommended below 2 years? Because it do not elicit any protective immune response less than 2 years. Now for all polysaccharide vaccine, this is the fact that polysaccharide specific T independent response is poorly developed in younger children due to immaturity. So when you are giving some polysaccharide vaccine, the T cell independent response is very poorly developed in less than 2 years. That's why it is not given in less than 2 years due to immaturity in the infant immune system characterized by lack of functional splenic marginal zone. It is recommended that all the adults who are more than 65 years of age older, they should receive 1 dose of this PPSV, that is polysaccharide vaccine for this pneumococcus 23 variant. Revaccination 5 years after the first dose is recommended for a person more than 2 years of age who have underlying the medical condition, but not routine for those who have only indication of more than 65 years. So usually they are given for the high risk patients. Capsular polysaccharide, as I told, these are 23 valent. Now because it is not given in less than 2 years and good number of neonates or the infants less than 2 years, they have infection due to strep pneumoniae causing meningitis. Okay, that's why the polysaccharide protein conjugate vaccine PCV 13 and PCV 7 were designed, and these vaccines, the biggest advantage is that they can be given in less than 2 years, and it was found that they help in reduction of invasive diseases. The polysaccharide protein conjugate vaccine PCV7 and PCV13, they induce T cell dependent immune response under 2 years and may reduce the pneumococcal transmission through a herd immunity. PCV13 licensed to replace PCV7 in 2010 for children between 6 weeks to 5 years, and it is approved by both USA and EU. Then the Neisseria meningitidis, it is also a gram negative diplococci. It is also having a polysaccharide capsule, and not exactly capsule, the cell wall has polysaccharide as well as it has a capsule of polysaccharide. Okay, so the monovalent and polyvalent vaccines are available. The vaccines against this capsular polysaccharide against group A, C, W135 and Y, these are available. These 4 groups are important groups: A, C, W135 and Y. These are available against this Neisseria meningitidis. They induce good immunity after single dose only, and same as pneumococcal vaccine, they do not produce immunity less than 2 years. Immunity is group specific. No group B vaccine is available. Okay. Immunologically, they are inferior to conjugate vaccine. They do not induce T cell independent immunity. They are hence ineffective in young children, inability to elicit long-term immunologic memory. They do not provide booster response. We need to give them booster doses, and they do not decrease the pharyngeal colonization. So Neisseria meningea, you all know, they colonize in the parents. That's why they help in transmission the disease. Okay. So here they do not decrease the pharyngeal colonization and induce phenomena of immune hyper-responsiveness. The current polysaccharide vaccine which are available, Meningivac, Menohuman, ACVac, ACW, Vivax. So these are Sanofi and GlaxoSmithKline. So they are targeting the group A, C and W130. For Neisseria meningitis also the conjugate vaccine is now available, just like the conjugate vaccine in pneumococcus, where it is conjugated with diphtheria toxoid that makes the vaccine immunogenic below 2 years. Okay. Meningococcal capsular vaccine, conjugate vaccine is mainly given in Africa. There are also conjugate vaccine targeting different other polysaccharides. The Men Africa Vac, it is capsular group A conjugate vaccine for Africa, reduce amination of group of polysaccharide, aldehyde and tetanus. These three components are there. So it is mainly exported to Africa country at 2010 and mass vaccination started, and there is seen that there is dramatic decline in the group A carriers because of this conjugate vaccine. Okay, that's why there is continuation of this vaccine. But one disadvantage you must remember of these vaccines, as I told, they target only one specific serotype. Yes, one or two specific serotypes which are there in that vaccine. So it was found that for all these polysaccharide vaccine, when we are administering, they usually decrease that particular strain, but the other strain, other capsular or non-capsular strain, they gradually emerge and they subsequently causes disease. This phenomena is known as replacement disease. For example, here, though the group A carriers has been declined, but gradually there is emergence of group C, W and X were found, and so you have to again target these vaccines. That's why they have revised trivalent MPS V3 and they reserve for capsular group C and W for the epidemics whenever some disease happens by these serotypes. These are the licensed vaccines for meningococcus. Usually safe, they are safe in pregnancy also, but sometimes they cause Guillain-Barré syndrome. Fine. The WHO recommendation for this Neisseria meningitis, when the endemic rate is very high, that is more than 10 per 1 lakh or intermediate 2 to 10 per 1 lakh, then these vaccination programs for meningococcus has been started. When it is less frequent, it is only given to the high risk. Group B, as in the beginning I told, there is no group B vaccine, but gradually it was targeted that, and they found that group B polysaccharide, they are antigenically similar to human neural tissues. The outer membrane vesicles are targeted for this group B vaccine by the method of reverse vaccinology. What is reverse vaccinology? You have to identify each and every component of the bacteria, and from reverse direction means from component you will find out that what all component are eligible to produce immune response. So suppose in the beginning you have 100 components and gradually you narrow down to 2 to 3 ideal components which can be introduced in terms of vaccine, which are potent candidates to cause immune response. This is known as reverse vaccinology because you are starting from the bacteria and ending in the immune response. Okay. So outer membrane protein 4A is a primary antigen, and here Neisseria heparin binding antigen C2 fragment involved in the endothelium. So no need to remember, you just remember that group B vaccine of Neisseria meningitis has been targeted for outer membrane vesicle by reverse vaccinology. In India also we have bivalent and quadrivalent, which has been given in Meghalaya and Tripura outbreaks. Okay, and the IAP program that recommends meningococcal vaccine for more than 2 years of age in certain high risk groups, especially military recruits and Haji pilgrims. Coming to the Hib vaccine, it is again same type like other capsulated vaccines. Hib PRP is indicated for this purified PRP found to be immunogenic. It is recommended more than 5 years, poorly immunogenic in less than 2 years. They are conjugated with protein carriers like diphtheria and toxoid or meningococcus outer membrane protein to increase the immunogenicity. Typhoid, we have 3 different types of vaccine. Typhoid, this TY21A, it is a attenuated vaccine where the S typhi has been attenuated. There is certain gene deletion, that is GAL-E gene deletion of this TY21A strain, and it can be given more than 2 years, but it only give partial protection to para typhi A, and it needs a booster dose. VI polysaccharide antigen, the subunit vaccine, is also available for typhoid. It also can be given for more than 2 years, but it also needs booster. There is wholesale killed vaccine TAB, and it gives a good protection, but it needs 2 doses. So these are the different thing, and this is the attenuated one, TY21. S typhi, the GAL-E gene has been deleted and it is a VI mutant. So once you take this vaccine, this because of this deletion, this attenuated strain, it develops or it multiplies for 4 to 5 generation, and after that there will be automatic death happens inside the body, so it cannot produce any disease. Okay, however, the efficacy of all type of typhoid vaccine varies from 2 to 3 years, around 60 to 70%. Pertussis, it is a whole cell kill vaccine. It is very effective. The detoxification of this whole bacterial cell done by 0.2% merthiolate for the storage. Pertussis vaccine usually administered with diphtheria and tetanus toxoid. Here the pertussis act as an adjuvant for toxoid producing better antibody response. So what do you mean by adjuvant? Adjuvant is a substance which allows the slow release of the antigen at the site. So in the vaccine DPT, D is diphtheria, P is the pertussis, and T is the toxoid. So pertussis component has been added as an adjuvant to diphtheria and tetanus toxoid because it helps in slow release of diphtheria and tetanus toxoid from the site, and gradually there will be better antibody response. These are the various targets for the pertussis: PT, FHA, FR and so these components can be taken from the pertussis and it can be combined with diphtheria and tetanus and prepare various acellular pertussis vaccine. The next, as I told, the Shigella E. coli, Vibrio. These are not routinely recommended as per the IEP program, but depending on the scenario, like whether there is some epidemic or outbreak, or you are traveling to like person from developed country, they are traveling to countries like India where there is a high chance of unhygienic food. So these conditions where you can or you are recommended to take these vaccines. So cigatoxin producing E. coli, cigatoxin toxoid has been prepared targeting the B component and AB subunit. The latter is more immunogenic, that is AB, but the STX2 toxin has been inactivated with formalin, and it is given in mice, and it is found 100% protection. So many of them, they are under trial or research, and very few has been marketed. In another trial, they have conjugated with Colorad and toxin adjuvant, and they are given intranasally, which found to be immunogenic. They have also targeted the O-specific polysaccharide conjugate vaccine, and it is detoxified O157 polysaccharide. The STEC strain were live attenuated vector vaccine, and these attenuated Vibrio cholerae vector Peru 2 strains which are having the STXB and the intimine are also targeted. Okay, so these are the various method how the cigatoxins are produced. Similarly, enterotoxigenic E. coli whole cell killed vaccine, live attenuated vaccine, purified antigen vaccine. So you no need to remember, however you just remember that what are the conditions where these vaccines are recommended. And one thing I want to tell you here, one component is edible vaccine. One type of vaccine is edible vaccine. How this has been produced? Because of the recombinant technology, these bacteria or the components were expressed in the edible part of the plant, and this can be edible. Okay, and there is immunogenicity. Similarly, cholera, there are basically two types, Dukoral and Orocol, which is live oral vaccine and whole cell kill vaccine. And there is also whole cell injectable vaccine. They usually target the Inava and Ogawa strains of the Vibrio, and injectable as well as oral dose are also available, but many of them they give only 60 to 80% of protection. I think, okay, one more. Diphtheria, the formal toxoid diphtheria toxin that has been inactivated, and they are adsorbed in aluminum phosphate, and they found to be more immunogenic. So DPT as a part of your national immunization program, they are given to the young children in 25 LF globular flocculation unit, and in older it is given in 1 to 2 flocculation unit. I think that's it. Any doubt, we can take some questions. Just wait, I want to finish the lecture. कोई है नहीं क्या? इसको बंद करना है थोड़ा LT वाले को। LT जो नहीं, ये जो चलाते हैं उनको बंद करना था।
वो अभी मैडम अभी आए नहीं है, आएंगे जब प्रोजेक्ट सीरियस है, इधर वाला अभी वो कुछ बोला नहीं, आए नहीं है, तो मैंने बोला आप करने चाहिए आप करिए,